Hello. Our topic for this unit now, we're going to kind of take that information that we learned about DNA replication in a previous week, and now we're going to talk about what happens in order to make RNA and protein within a eukaryotic cell. Okay? So you may remember from a previous video, I put up the flow of genetic information. All right? And we've already covered DNA can make an exact copy of itself via DNA replication. And it needs to make a copy of itself before it can undergo mitosis to generate a new cell, because that new cell needs a copy of the instruction manual, just like the old cell had. Okay? So once our DNA has replicated and we've got our new cell, that new cell is going to want to start making proteins. And in order to make proteins, it actually has to make an intermediate product. All right? It has to make RNA. And the reason for that needing to happen, all right? If we go here, this is going to be our nucleus. Okay? This is going to be the cytoplasm of our cell. All right? Within our nucleus, oh, that's terribly squeaky, I apologize. All right, at least you'll be awake. All right, in the nucleus, remember DNA has to stay here in the nucleus. It can't move outside into the cytoplasm. So the DNA replication is going to occur in the nucleus. But we have to have a way to get that information from the nucleus out to the cytoplasm, okay? Because out in the cytoplasm, this is where translation happens, all right? And that process of translation is making our protein, all right? And the cellular structure that's going to be involved in that, just to remind you, is going to be our ribosome, because our ribosomes exist out in the cytoplasm. So we have to have a way for the information from the DNA to get out to the cytoplasm. And that's why we have the intermediate RNA. Okay? Because RNA molecules can leave the nucleus via those nuclear pores. It can travel out to the cytoplasm where it can be used for translation. Okay? So our process here, we've got DNA that's going to encode RNA via the process of transcription. And that process of transcription is going to occur oh goodness, I can spell. I promise I can spell. Just not today. Is going to happen in our nucleus. All right? The RNA, the information from the RNA is then going to be translated to protein. And that process of translation is going to occur out in the cytoplasm. Okay? So RNA really kind of represents a unique intermediary and is the way that the cell deals with having partitioned off its DNA inside a membranous organelle inside of the cell. Okay? So that RNA then is going to be what carries the genetic information to the cytoplasm for translation to happen. All right? And that RNA molecule, okay, is going to be very similar in structure to our DNA molecule. And I'm going to draw a little one up here. Okay? And at this point, this should look fairly familiar to you because I've drawn this a lot. All right? Now, what makes this an RNA molecule, We've got our hydroxyl group at that 2' carbon. We've got a hydroxyl group at the 3' carbon. Okay? That sugar is going to be different. The sugar in RNA is a ribose sugar. The bases in RNA are also different from DNA. Our bases in RNA are going to be adenine, uracil, cytosine, and guanine. Okay, those are going to be the 4 bases that are going to be used in an RNA molecule. All right. So if I put up a sequence that has a uracil in it, you automatically know this has to be an RNA molecule, okay? And our RNA molecules are always going to be synthesized on that 5' to 3' direction. So they're always going to run 5' to 3', okay? So that makes that a little bit simpler as compared to DNA, all right? So that sequence then, if you see a U in it, you know that sequence I'm giving you is an RNA sequence, all right? Now, if I ask you to transcribe a sequence, okay? For example, this is going to be our sequence, okay? And this is going to be our DNA sequence, okay? And in this case, this is going to be 5', this is going to be 3' here. To transcribe this, all right, the base pairing in RNA, if you see an A, you're going to insert a U, okay? So if you see a T, it'd be an A. C and G work the exact same way as in DNA, okay? T would give you an A would give you U, C would give you G, okay? So this would be our RNA, oops, I forgot the R there, our RNA molecule, okay? So if I'm asking you to transcribe, this is what I'm asking you to do. I'm asking you to take the DNA sequence and tell me what the RNA sequence for that DNA sequence, what it would be, okay? So with our RNA, it's going to carry the information from the nucleus to the cytoplasm so that we can talk about translation. Before we get into translation, though, we're going to talk in a little more detail about how transcription works. And that's going to be the next topic for our video. All right. Thank you for your attention. If you have any questions, please let me know.